Okay, so hello again, guys. Today we're going to talk about what is known as cell division or mitosis. Um, in the cell cycle, we have two stages. We have the interphase and we have the mitosis. Now, before, why a cell needs to divide? We said in the previous video that sometimes when we go to the beach in summer and we have a form of tan, we lose some skin cells. Or, for example, when we cut our fingernails, they grow back again. Or when we have a wound or an injury, we notice that it will heal after time and it will be covered. This means that our cells are multiplying, they're forming new cells. How? By something known as the cell division. Now, before going into details of what a cellular division is, I'm going to watch a small video with you guys about mitosis, and then we will continue.
Cell division is required for an organism to grow, mature, and maintain tissues. During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. Mitosis is only one part of what is called the cell cycle. For many eukaryotic cells, a cell is duplicated every 24 hours. Most of the life of a cell is spent in interphase. Interphase consists of three stages called G1, S, and G2. G1, or gap1, is the first growth stage of interphase. In G1, the cell grows to nearly its full size and performs many of its specific biochemical functions that aid the organism. Next is the S, or synthesis phase. This is an important stage because it is during the S phase that DNA in the nucleus is replicated. The cell next enters another growth stage called G2, or gap2. It is during G2 that the cell finishes growing. Once the cell has duplicated DNA in the nucleus and two centrosomes have appeared in the cytoplasm, mitosis can begin. For a typical eukaryotic cell, this will last about 80 minutes. During the first stage of mitosis, called prophase, we first see the classic chromosome structure. This occurs through a condensation process.
These are the chromosomes you
At the
see.
same time, protein strands called microtubules appear from the centrosomes in animals. Finally, a structure found within the nucleus, the nucleolus, disappears. Next,
I want to mention something here, that the centrosomes are presented only in an animal cell because we are going to take the plant cell in which these centrosomes are not presented. So we notice that mitosis occurs in plant cells as well, but with the absence of these centrosomes.
prometaphase begins when the nuclear membrane is broken down. At the same time, microtubule strands, or spindle fibers, are growing from the centrosomes.
Like the threads, you see?
These strands attach to a protein structure called the kinetochore. One kinetochore is attached to the centromere of each sister chromatid. Next comes metaphase. During this stage, the sister chromatids align along the center of the cell so that both chromatids face toward opposite poles of the cell.
So in the
Now
middle,
the sister chromatids are ready to be separated. This occurs during anaphase through a shortening of the microtubules attached to the kinetochores. Additionally, the poles of the cell move farther apart. causing increased separation of sister chromatids. At the end of anaphase, the sister chromatids have moved to the two ends of the cell. Telophase is the final stage of mitosis. It is here the components of the new cells begin to appear. At this point, the spindle fibers are broken up. A new nuclear membrane surrounds the chromosomes at the end of each cell, and the chromosomes uncoil and return to an uncondensed state. Mitosis is now complete. The formation of two cells is all that remains. Following mitosis, the cell undergoes a process called cytokinesis. First, the cell is compressed by a contractile ring that divides the cell in nearly equal halves. By now, the organelles in the cell have been replicated and are now divided between the two halves of the cell. This includes mitochondria, Golgi bodies, and the rough ER. Plant cells also have chloroplasts. Once split, the two new cells are now fully in the G1 stage of interphase and ready again to begin their growth. Let's watch the process one more time. Mitosis begins with prophase. Notice the DNA condensing into chromosomes during this stage. Microtubules appear during prometaphase, and the nuclear membrane breaks down. Metaphase occurs when the chromosomes are aligned at the center of the cell.
Metaphase, middle, in the middle.
During anaphase, the chromosomes are moving apart.
Or away, or apart. Yes.
The telophase stage is marked by the appearance of new nuclear membranes. This is the end of mitosis. Finally, the splitting of the cell occurs during cytokinesis. The two new cells are now ready to grow and perform their specialized functions.
And these two daughter cells will undergo mitosis again after entering interphase. And it is called, why is it called a cell cycle? Because it happens all the time. So one cell divides to give two daughter cells, as you see here. These two daughter cells will undergo mitosis again to give four daughter cells. The four will divide, and so on. So to return to our explanation, we said that the cell cycle starts with interphase, which is the first stage. Preparing to any division, the cell duplicates its genetic material in order to conserve the genetic material of this species. So in this mother cell, we notice that we have here the genetic material, which is the DNA or the chromosomes presented in the nucleus. They are going to be what? Duplicated, and that is the main purpose behind interphase. It allows the duplication or the replication of the genetic material. You notice here one chromatid changes into two chromatids. Okay, after the interphase, we go into the details of mitosis, which is made up of four main phases. We have the PMAT, it's known as the PMAT. PMAT, P for prophase, M for metaphase, A for anaphase, and T for telophase. Let's go into details and see what happens in each phase of these. Now, all the eukaryotic cells, they undergo a division that increases their number without losing or gaining any genetic material. That's the idea behind mitosis, that the genetic material is conserved. It does not change. It's the same. A cell containing 46 chromosomes will divide. It will give two daughter cells, each containing 46 chromosomes, and not the half or a quarter or not, no. Because when it gives half, we go into another names, which is like meiosis, and we're going to talk about this later on. But when we say mitosis, it means I do have a cell containing 4 chromosomes. It will end up dividing, giving two daughter cells, each containing the same number as this one here, okay? This division is called mitosis, and we said it consists of 4 different stages, starting with the prophase. During prophase, as you notice, the nuclear membrane starts to disappear, and we have the formation of spindle fibers, as you see here, that are going to grow up, as we saw in the video, in order to be attached to the chromosomes. Now then, we have the metaphase. And metaphase, the word reminds me of middle. For that reason, when I was a student in grade 9, I remember that metaphase, so chromosomes stand in the middle of the cell. We call it the equator of the cell. They line up at the equator. Okay, during anaphase, A for away, the sister chromatids of a chromosome are going to be away from each other. They split, and each sister chromatid will be separated from the other, as you see here. Then, after this, we have the telophase. During telophase, as you notice, the nuclear membranes appearing again, and the cell is starting to what? To be divided into two, which is known as cytokinesis. We have two identical daughter cells, as we notice here, each containing, if we count the chromosomes 1, 2, 3, 4, the same number as the one presented in the original cell. For that reason, we say mitosis is a process that conserves that genetic material as a number. We start with 4 chromosomes, we end up with 4 chromosomes. We start with 8, we end up with 8. We start with 46, we end up with 46. Now, this graph represents the quantity of the genetic material during time of a cell cycle. It shows the phases of a cell cycle. And as we said before, the cell cycle is made up of interphase followed by mitosis. Mitosis, which is made up of P, mat, P for prophase, M for metaphase, A for anaphase, and T for telophase. Let's answer the questions followed by this graph. Put a title for the given graph. So this title would just said that it shows the quantity of the genetic material in arbitrary units as a function of the time in hours. So we said that time is in hours. It needs about the cell cycle they said in the video, about 24 hours, sometimes less, sometimes more. Now, what is the scale on which the graph was drawn? So as we notice here, we have on the y-axis 0.4 arbitrary units, and here we have 1 for each 1 hour, for each division. Yes, determine the duration of each phase of the cycle. So if we talk about the interphase, we notice that it starts at time 0, it finishes at time equals 12 hours, so it needs about 12 hours to happen. Whereas mitosis, it starts at 12 and it finishes the telophase at 13.5, so it needs 13.5 minus 12, which is 1.5 hours. Now, if we're going to take each one by itself, we notice that gap 1, which is the first stage in the interphase, it takes 5 hours, whereas the synthesis phase, it takes 8 minus 5, which is 3 hours. The G2 is about 4 hours, and we can notice that all of the mitosis is 1.5 hours. Now, what is the event that takes place during interphase? What happens mainly during interphase? Why does it need to occur? The genetic material, the main purpose of interphase is to duplicate the genetic material. If you notice here, it was 0.4, it changed to 0.8, so it's duplicating. This is happening in the synthesis. It's increasing. The quantity of genetic material duplicates here. It was 0.4, it became 0.8. And that is the main purpose of interphase. Okay, tabulate the graph, so change it into a table. This will be a homework for my students. If the cell at time 0 was a zygote, how many cells are produced at time 13.5? So, if we have a zygote here and it's undergoing interphase, then 1 mitosis, how many cells will be produced at 13.5? So, after 1 complete interphase and mitosis, we will have 2 daughter cells. If this zygote undergoes 5 mitotic divisions, so imagine you guys, 1 cell is producing 2, 2 are producing 4, so each one is producing another 2, and then the 4 will undergo mitosis to produce cells again. So if this happens 5 times, how many cells are going to be produced? It's as if 2 to the power 5. Okay, it's a math issue. So 2 to the power 5 is 32. We have 32 cells being produced. Schematize the chromosome seen under at the 4th and the 11th hour. So at the 4th hour, which is here, the genetic material is not duplicated, so it's made up of, remember, of 1 chromatid only. Whereas at the second one, which is 11, no, the chromosomic material is duplicated, so it's made up of two chromatids. This could be seen here.